Before Anomalocaris, nothing on Earth had eyes designed to find prey. Nothing had claws designed to catch it. Nothing had a mouth designed to crush it. Every adaptation for predation that has ever existed in the history of animal life, the hawk's talons, the shark's teeth, the wolf's speed, traces back to innovations that began in the Cambrian Ocean 500 million years ago. And the animal that started all of it looked like nothing you have ever seen. And it was enormous. And it was everywhere. Before we get down to business, please subscribe to this channel. Your support really helps us to continue this work. Thank you. To understand what Anomalocaris was and why it matters, you first need to understand the world it entered. And that world was, by any modern standard, profoundly strange. Not strange in the way that deep ocean ecosystems are strange, exotic, but comprehensible, operating by the same ecological rules we recognize everywhere else. Strange in a more fundamental sense. Strange because the rules themselves had not yet been written. Because the ecological relationships that govern every living community on Earth today, predator and prey, hunter and hunted, the entire architecture of fear and pursuit and escape that structures animal behavior from the smallest invertebrate to the largest mammal, none of that existed. The ocean was full of life, and nothing in it was afraid. The Ediacaran period, which immediately preceded the Cambrian and ran from roughly 635 million to 539 million years ago, was the first era of complex multicellular animal life on Earth. The organisms that populated Ediacaran sea floors were genuinely extraordinary. Flat, frond-like creatures, disc-shaped organisms, quilted mattress-like forms that have resisted confident classification for decades because they were so unlike anything that followed them. Dickinsonia, which could grow to more than a meter in length, was a ribbed oval of biological material that moved slowly across the sea floor, absorbing nutrients through its base. Charnia was a frond anchored to the sea floor like a sea pen, filtering whatever drifted past. Tribrachidium was a disc with a threefold symmetry found nowhere in the modern animal kingdom. These organisms, and the dozens of other forms found in Ediacaran deposits worldwide, from the Mistaken Point cliffs of Newfoundland to the Flinders Ranges of South Australia, shared one remarkable characteristic. They show almost no evidence of predation, no bite marks, no repair scars where a wound had healed, no shells or hard parts whose obvious function would be protection against attack. The Ediacaran world was, as far as the fossil record tells us, a world without predators, a world without fear. This does not mean Ediacaran life was simple or static. It was diverse. It was globally distributed. It had been evolving for tens of millions of years and had developed a range of body plans and ecological strategies. But the ecological strategy of actively hunting and consuming other animals, of predation in the strict sense, appears to have been absent or vanishingly rare. Some researchers have proposed that certain Ediacaran organisms may have been early predators, and there are trace fossils from the late Ediacaran that could be interpreted as predatory behavior. But the overwhelming character of Ediacaran ecology, as preserved in the fossil record, is one of passive feeding, of filter feeding and mat grazing and osmotrophic absorption, of organisms exploiting resources that did not resist being exploited. It was a world of victims with no predators, which means, technically, they were not victims at all. They were simply organisms living in an ocean where the concept of being eaten had not yet been operationalized by evolution. And then, beginning approximately 539 million years ago, everything changed. The change was not gradual. It was not a slow accumulation of innovations spread over hundreds of millions of years. By geological standards, it was essentially instantaneous, compressed into a window of perhaps 20 to 25 million years, possibly less, that geologists call the Cambrian explosion. In that interval, the fossil record goes from the soft, slow, predator-free world of the Ediacaran to something that looks, in its basic ecological architecture, recognizably modern. Animals with eyes, animals with legs, animals with shells and claws and teeth, and the behavioral repertoire of hunter and hunted. The full complexity of a predator-prey structured ecosystem appearing in the rock record with a suddenness that Darwin himself found deeply troubling because it seemed to contradict his model of gradual evolutionary change. The Cambrian explosion is one of the most studied and most debated events in the history of life, and a full accounting of its causes and mechanisms would require its own documentary. But for our purposes, the critical point is this. Among the innovations that appeared in the Cambrian, perhaps the most ecologically transformative was predation. The active, directed, behaviorally sophisticated pursuit and consumption of other animals. And among the first and most spectacular practitioners of this new ecological strategy was the animal we are here to discuss. An animal whose body plan was so alien to anything in the modern world that when its fossils were first discovered, scientists spent decades misidentifying its individual body parts as separate organisms, unable to conceive that something so large, so anatomically complex, and so clearly designed for killing could have existed at the very dawn of complex animal life. The story of Anomalocaris' discovery is one of the most entertaining detective stories in the history of paleontology, and it deserves to be told in some detail because it illustrates both the difficulty of reconstructing ancient life from fragmentary fossil evidence and the degree to which our preconceptions about what ancient animals should look like can lead even expert scientists systematically astray. The story begins in 1886 in the mountains of British Columbia, Canada, where a geologist named Richard McConnell collected a fossil from Cambrian age rocks and sent it to the Geological Survey of Canada. The fossil was a curved, segmented appendage, roughly the size and shape of a large shrimp, and the paleontologist who described it, Joseph White Aves, interpreted it as exactly that. A shrimp, or more precisely, the abdomen of a shrimp-like crustacean, lacking its head. He named it Anomalocaris canadensis. The name means abnormal shrimp from Canada, which was a reasonable interpretation given what he could see, and a wonderfully ironic name given what Anomalocaris actually was. 2 decades later, Charles Doolittle Walcott, the secretary of the Smithsonian Institution and one of the most important paleontologists of the early 20th century, discovered the extraordinary fossil deposit in the Canadian Rockies that would become known as the Burgess Shale. The Burgess Shale, which Walcott began collecting systematically in 1909, is one of the most important fossil localities ever found, a deposit of Middle Cambrian age in which the soft tissues of organisms were preserved with a fidelity that is almost without parallel in the fossil record. In a normal marine environment, soft tissue decays within days to weeks of an organism's death, leaving only hard parts, shells, bones, teeth, teeth to be fossilized. At Burgess, some combination of rapid burial, low oxygen bottom conditions, and sealing by fine-grained sediment preserved not just hard parts, but the complete anatomy of soft-bodied organisms, their muscles, their digestive systems, their gills, their eyes. Walcott collected tens of thousands of specimens from the Burgess Shale over multiple field seasons, and among them were fossils of the circular, tooth-ringed mouth of a large animal, which he interpreted as a jellyfish of some kind, and what appeared to be the remains of a large, soft-bodied organism, which he did not fully understand. He did not recognize that these were parts of the same animal. He did not recognize that the shrimp-like Anomalocaris appendage described by White Eaves was also part of the same animal. 3 different fossils, 3 different animals. That was the interpretation that stood for most of the 20th century. The revision came in 1985, when Harry Whittington and his colleagues, Simon Conway Morris and Derek Briggs, working on a major reassessment of the Burgess Shale fauna, recognized that the circular mouth fossil, which had been named Peytoia, and the appendage fossil of Anomalocaris, and several other fragmentary specimens, were all parts of a single large predatory animal. The appendages were not shrimp abdomens. They were the grasping frontal appendages, the claws, of a large arthropod-like creature. The circular toothed structure was not a jellyfish. It was the mouth. And the complete animal, when reconstructed from all the fragments, was unlike anything anyone had expected to find in the Cambrian. It was large, it was fast, it was anatomically sophisticated, and it was clearly, unmistakably, a predator. In the years since that reassessment, additional specimens from multiple locations worldwide have dramatically refined our understanding of Anomalocaris and its relatives. The most complete and informative specimens have come from the Chengjiang fauna of Yunnan province in southern China, a deposit of early Cambrian age that is slightly older than the Burgess Shale and that has produced exquisitely preserved radiodont fossils, including complete animals with their soft tissues intact. From these specimens, from continued work on Burgess Shale material, and from newer discoveries in sites ranging from Morocco to Greenland to Australia, we now have a picture of Anomalocaris that is detailed enough, something meaningful about how it lived, how it hunted, and what it meant for the history of life. So, what was it exactly? Anomalocaris was a radiodont, a member of an extinct group of animals that were related to, but distinct from, the arthropods that would eventually give rise to the crustaceans, insects, spiders, and millipedes that dominate the modern world. It had a body that was roughly streamlined, perhaps 0.5 meter to 1 meter in length, depending on the species and specimen, though some estimates based on fragmentary evidence push certain species somewhat larger. Along the sides of its body ran a series of lateral lobes, flexible fin-like flaps that it undulated in a smooth wave motion to propel itself through the water, in the same general way that a modern cuttlefish or a flattened ray uses lateral fins for locomotion. This form of locomotion, called labriform or undulatory fin swimming, is remarkably efficient and maneuverable, allowing precise 3-dimensional control of movement in ways that simple tail-flicking swimming does not. Anomalocaris was not a clumsy bottom crawler. It was an active, maneuverable swimmer capable of pursuing prey through open water. At the front of its body, 2 large articulated appendages extended forward, each composed of multiple segments with spines on the inner margins. These frontal appendages are the structures that were originally misidentified as shrimp abdomens, and in their full context, they are clearly grasping organs, the equivalent of claws or hands, designed to seize prey and bring it toward the mouth. Below the frontal appendages, or more precisely at the anterior end of the ventral surface, sat the mouth, a circular opening surrounded by a ring of plate-like teeth, hard enough to be preserved in the fossil record, arranged in a pattern that could close inward to crush whatever the frontal appendages had delivered to it. The entire feeding apparatus, frontal appendages plus circular crushing mouth, was an integrated predatory system of a sophistication that had no precedent in the history of life. Nothing before Anomalocaris had been built with such obvious anatomical commitment to the business of catching and processing other animals. And then there were the eyes. The eyes of Anomalocaris are among the most remarkable structures in the Cambrian fossil record, and their significance goes beyond the merely anatomical. Preserved specimens from the Chengjiang fauna show the eyes of Anomalocaris as large, stalked structures positioned at the front of the head, and in exceptional specimens, the internal structure of the eye is preserved well enough to count the individual optical units, the ommatidia, that made up the compound eye surface. The count is extraordinary. Some specimens preserve eyes with more than 16,000 individual optical units, each one a separate lens contributing to a wide-field, high-resolution image of the surrounding environment. For comparison, a modern housefly has approximately 4,000 ommatidia. A modern dragonfly, which has among the most sophisticated eyes of any living insect, and which is itself a highly effective aerial predator, has up to 30,000. Anomalocaris, 500 million years ago, had eyes that were within the same order of magnitude as a modern dragonfly, and it had them in an ocean where nothing else had eyes of comparable sophistication. This is worth pausing on, because the implications are profound. Eyes of the complexity preserved in Anomalocaris are not simple light detectors. They are image-forming organs. They create a detailed, spatially resolved picture of the environment, capable of detecting the movement, shape, and position of objects in the visual field, with enough precision to support directed, targeted predatory behavior. An animal with eyes like those of Anomalocaris could see its prey, could identify it, could track its movement, could calculate an interception trajectory. This is a level of sensory capability that is not just quantitatively greater than what preceded it, but qualitatively different. Before compound eyes, marine animals could detect light and dark, could respond to the shadow of an approaching object, could orient toward or away from a light source. After compound eyes, an animal could see the world as a spatially organized scene, populated by other organisms whose size, speed, and direction of movement could be assessed and responded to appropriately. The evolution of the image-forming eye was not just a sensory advance. It was a cognitive advance, enabling a sophistication of behavioral response to environmental information that was entirely new in the history of animal life. And here the story takes its first major unexpected turn, one that has significant implications for how we understand Anomalocaris as a predator. For decades after the reconstruction of Anomalocaris from its fragmentary remains, the standard interpretation was that it was an apex predator that used its spiny frontal appendages to capture trilobites and other hard-shelled Cambrian organisms, and its circular toothed mouth to crush their shells. The visual of Anomalocaris as a shell-crushing monster pursuing armored trilobites through the Cambrian Ocean became canonical, reproduced in textbooks and museum exhibits, and popular science articles worldwide. It was a compelling and logically consistent narrative. Here is the first predator. Here are the first animals with hard protective shells. Here is the evolutionary arms race that drove Cambrian diversification. The problem is the teeth. In 2015, a detailed biomechanical analysis of the oral plates of Anomalocaris was published, examining the structure, material properties, and range of motion of the tooth ring elements, with sophisticated engineering tools that had not been available to earlier researchers. The conclusion was unexpected and, to many paleontologists, initially uncomfortable. The oral plates of Anomalocaris canadensis were not rigid enough to crush the mineralized shells of trilobites or other hard-shelled Cambrian organisms without fracturing. The plates had some hardness, enough to handle soft or lightly mineralized prey, but not the structural rigidity required to generate the bite forces needed to crack a well-mineralized trilobite exoskeleton. The implications for Anomalocaris's diet were significant. If it could not crush hard shells, what was it eating? The current leading hypothesis, supported by the biomechanical evidence and by what we know about the structure of the Cambrian ecosystem, is that Anomalocaris canadensis was a predator of soft-bodied or lightly armored prey. The Cambrian Ocean was full of soft-bodied animals, worms of various kinds, early chordates, poorly mineralized arthropods, organisms that had not yet developed the defensive hardparts that later became so common. A large, fast-swimming predator with excellent vision and grasping appendages capable of seizing and manipulating soft prey would have been devastatingly effective against such organisms, without needing to crush hard shells at all. Anomalocaris may have been less like a modern shark and more like a modern large cephalopod, a sophisticated, visually guided, actively pursuing predator of soft prey, dominant not because of crushing power, but because of speed, maneuverability, and sensory sophistication. Some specimens of Anomalocaris-like animals have also been associated with coprolites, fossilized feces, that contain the fragmentary remains of trilobites, which complicates the picture somewhat and suggests that at least some radiodont species may have been capable of processing harder prey than the biomechanical models suggest. The honest answer is that Anomalocaris's diet is still an active area of research, and the story is more complex than either the original shell crusher hypothesis or the revised soft prey model fully captures. What is not in dispute is that Anomalocaris was a visually guided, actively pursuing, anatomically sophisticated predator, the most complex predatory organism that had yet existed on Earth, operating in an ecological context that had never before included anything remotely comparable. The effect of Anomalocaris and its radiodont relatives on the evolutionary trajectory of Cambrian life is difficult to overstate, because the appearance of an effective predator in a previously predator-free ecosystem is not just a local ecological event. It is a global evolutionary signal transmitted to every prey organism in the system simultaneously, and the response to that signal drives evolutionary change at a rate and in a direction that would never have occurred without it. Think about what it means to be a prey animal in a world where predation is a new phenomenon. You are an organism whose ancestors, for hundreds of millions of years, faced no directed threat from other animals. Your body plan was optimized for other challenges, for finding food, for reproducing, for competing with other members of your own species for resources. You had no armor because armor was never necessary. You had no escape behavior because there was nothing to escape from. You had no capacity for fear in any meaningful neurological or behavioral sense, because the stimulus that generates fear, the presence of a predator capable of consuming you, had never existed in your evolutionary history. Now, it exists. Now something is hunting you. And the organisms in that Cambrian ocean that happened, by random genetic variation, to be slightly harder to catch, slightly better protected, slightly more capable of detecting and responding to the approach of a predator. Those organisms survived at slightly higher rates than their less fortunate neighbors. They reproduced. Their offspring inherited and built on their defensive advantages. Generation after generation, in the relentless arithmetic of natural selection, the prey community was shaped by the predator, pushed toward harder shells, faster escape responses, better sensory systems for detecting threat, behavioral repertoires that included something functionally equivalent to fear. And simultaneously, the predator community was shaped by the prey. Prey that became harder to catch created selective pressure for more effective capture mechanisms, better eyes, stronger appendages, more sophisticated hunting strategies. The predator and prey communities evolved together, each driving the other toward greater complexity in the process that evolutionary biologists call a co-evolutionary arms race. This is the engine that drove the Cambrian explosion, or at least a significant portion of it. Not just the appearance of predators, but the dynamic interaction between predators and prey. The reciprocal evolutionary escalation that forced both sides of the relationship toward greater anatomical and behavioral sophistication at a pace that passive, non-predatory ecological systems could never generate. The trilobites that dominated Cambrian seas were not simply opportunistic animals that happened to develop compound eyes and calcified exoskeletons. They were organisms under predatory pressure, and the compound eyes and calcified exoskeletons were the evolutionary response to that pressure. The diversity of Cambrian body plans, the extraordinary variety of forms preserved in the Burgess Shale and the Chengjiang fauna, and the dozens of other Cambrian Lagerstätten that have been discovered around the world, is in large part a record of the evolutionary arms race that Anomalocaris and its relatives initiated. To appreciate the full magnitude of what Anomalocaris set in motion, it helps to look closely at specific animals from the Cambrian fossil record and trace the exact nature of the evolutionary response to predation pressure. Because the arms race was not abstract. It was written in specific anatomical structures, in specific behavioral innovations, in specific ecological relationships that we can read in extraordinary detail from the Burgess Shale and Chengjiang deposits. The fossil record of the Cambrian is not just a catalogue of strange forms. It is a detailed archive of an evolutionary war, and the weapons and armor and tactics developed in that war are visible in the rock with a clarity that is genuinely astonishing. Trilobites are the most obvious place to start because they are the most abundant and best studied animals of the Cambrian fossil record, and because the evidence of predatory pressure on their evolution is both direct and dramatic. Trilobites were arthropods, distant cousins of modern crustaceans and insects, and they dominated Cambrian seafloors with a diversity and abundance that has no modern equivalent. More than 20,000 species of trilobite have been described from the fossil record spanning the Cambrian through the end Permian, and the earliest Cambrian trilobites were already mineralized, already armored with a calcified exoskeleton that was one of the first hard skeletal structures to appear in the fossil record of complex animals. The timing is not coincidental. The earliest trilobites appear in the fossil record at almost exactly the same time as the earliest evidence of predatory animals. Hard shells and predators arrive together in the Cambrian because they are causally connected. The shell is the response to the predator. But the shell was only the beginning of the trilobite defensive repertoire. As the Cambrian progressed and predatory pressure increased, trilobites evolved an extraordinary array of additional defensive structures and behaviors that are preserved in exquisite detail in the fossil record. Some trilobite species developed enormous spines projecting from their lateral margins, making them physically difficult to swallow and potentially painful to bite. The spines of Cambrian trilobites like Wallaceurops and the later Selenopeltis are so elaborate, so energetically costly to produce, and so obviously non-functional for any purpose other than predator deterrence, that they constitute direct fossil evidence of the intensity of predatory pressure on the trilobite lineage. You do not evolve a spine 3 times the length of your body unless something is regularly trying to eat you. More dramatic still is the evidence for enrollment behavior in trilobites. The ability to curl the body into a tight ball with the calcified exoskeleton, forming a complete defensive sphere around the vulnerable, soft underparts. Enrolled trilobites are found throughout the fossil record, and the enrollment mechanism, a complex system of interlocking skeletal plates that allows the animal to lock itself into a ball and resist being unrolled by a predator, became increasingly sophisticated through the Paleozoic. Modern pillbugs, which are crustaceans and therefore distant relatives of trilobites, use an analogous enrollment defense. The behavioral and anatomical solution is so effective that it has been independently evolved multiple times, which is itself evidence of how powerful the selective pressure of predation was and continues to be. Some enrolled trilobite fossils have been found with apparent bite marks on their outer surface, direct evidence of predator-prey interaction preserved in stone, a record of an attack that the trilobite survived by rolling into a ball, and of which the trilobite's exoskeleton still bears the marks. The eyes of trilobites are themselves a response to predatory pressure, or more precisely, a response to the predator-prey structured ecosystem in which early detection of threat was survival critical. Trilobite eyes were compound eyes, the same basic optical technology as the eyes of Anomalocaris, and some Cambrian trilobite species possessed eyes of remarkable sophistication. Certain trilobites in the family Phacopoida developed schizochroal eyes, in which each individual lens was separated from its neighbors by thick integument and had its own cornea, a design that has been shown to minimize spherical aberration and produce sharp images over a wide field of view. Other trilobites had holochroal eyes with thousands of closely packed lenses, providing wide-angle coverage of the surrounding environment with maximum light gathering. Still others had hypostomal eyes positioned below the head to watch for threats approaching from below. The diversity of Trilobite eye designs is itself a record of the different threat landscapes that different Trilobite species inhabited, each eye design optimized for the particular predatory environment its owner faced. The Cambrian arms race was not limited to Trilobites and their response to Anomalocaris. It played out simultaneously across dozens of other animal groups, producing a cascade of evolutionary innovations that transformed the biology of the Cambrian Ocean in just a few million years. The small shelly fauna, a diverse assemblage of mineralized microfossils that appears in the rock record just before the main Cambrian explosion, represents the very earliest response to predatory pressure in the animal kingdom. These tiny conical shells, tubes, plates, and spicules, produced by a variety of organisms whose precise identity and relationships are still incompletely understood, represent the first widespread mineralization event in animal history. Something triggered dozens of different animal lineages to independently evolve the ability to secrete calcium carbonate or calcium phosphate hard parts at approximately the same time. The most parsimonious explanation is that something started eating them. The small shelly fauna is, in all probability, the first wave of defensive armor in the history of animal life, predating even the trilobites, produced in direct evolutionary response to the first appearance of predatory animals in the late Ediacaran and earliest Cambrian. The Burgess Shale organism Wiwaxia is a particularly interesting example of defensive evolution because its armor was constructed from a completely different material and by a completely different mechanism than the calcium carbonate shells of most Cambrian animals. Wiwaxia was covered in overlapping carbonaceous scales called sclerites, forming a flexible coat of armor similar in principle to fish scales. And from this, armor projected a series of long, sharp spines that would have made it extremely unpleasant for any predator to attempt to swallow. The sclerites and spines of Wiwaxia are preserved in sufficient detail in Burgess Shale specimens to show that they were regularly shed and replaced as the animal grew, a sophisticated biological maintenance system for what was essentially a living suit of armor. Wiwaxia's relatives, the mollusks, would go on to develop calcium carbonate shells as their primary defensive structure. But Wiwaxia itself went a completely different direction, producing an organic armor system that was unique in the animal kingdom, and that stands as evidence of how many different evolutionary solutions the same selective pressure, predation, can generate simultaneously. Picaia graculans is perhaps the most personally significant organism in the Burgess Shale from a human perspective, because it is one of the earliest known chordates, a member of the animal group to which we belong, and its presence in the Burgess Shale alongside Anomalocaris tells us something important about the evolutionary context in which our own lineage originated. Picaia was a small, laterally compressed ribbon-like animal, perhaps 4 to 6 centimeters in length, that swam by undulating its body in the way that modern lancelets do. It had a notochord, the stiffening rod that is the defining feature of the chordate group, and that is the evolutionary precursor of the vertebrate spine. It had paired tentacles at its head end and a series of repeated muscle blocks along its body. It was, in short, a very early and very small chordate, and it was living in an ocean dominated by Anomalocaris. It was almost certainly prey. The survival of the chordate lineage through the Cambrian, given its apparent vulnerability as a small, soft-bodied, relatively slow swimmer in an ocean with large, fast, well-armed predators, is something that can only be explained by some combination of cryptic behavior, habitat choice, and the simple probabilistic reality that predators cannot catch every individual of every prey species. But the selective pressure of being a chordate in a Cambrian ocean with Anomalocaris was intense, and it drove the evolution of the chordate body plan in directions that were specifically adaptive for predator avoidance. The muscular body that allows rapid burst swimming. The streamlined shape that minimizes hydrodynamic drag during escape. The dorsal nerve cord and the sensory systems it served, providing the neurological capacity for rapid detection of and response to threat. All of these features, which are the defining characteristics of the vertebrate body plan as it emerged from its chordate ancestors, can be understood as adaptations for surviving in a predator-structured marine ecosystem. We are fast and smart and aware of our environment, partly because our Cambrian ancestors had to be, in order to avoid being eaten by Anomalocaris. The trace fossil record of the Cambrian adds yet another dimension to the arms race narrative, because behavior leaves traces even when bodies are not preserved. Cambrian sediments are rich in trace fossils, the tracks, burrows, and feeding traces left by animals moving through and processing the seafloor sediment. The pattern of trace fossils across the Ediacaran Cambrian boundary is striking. Ediacaran trace fossils are simple, shallow, and sparse, reflecting an ecosystem in which burrowing was not a particularly important behavioral strategy. Cambrian trace fossils are dramatically more complex, deeper, more varied, and more abundant, reflecting an ecosystem in which burrowing into the sediment had become a critical survival strategy. The reason is predation. An organism that can burrow rapidly into the sediment in response to the approach of a predator is significantly less likely to be eaten than one that remains on the sediment surface. The Cambrian burrowing revolution, the dramatic increase in the depth and complexity of animal burrows at the beginning of the Cambrian, preserved in the rock record as the Cambrian substrate revolution, is another direct consequence of the appearance of predation. The sediment became a refuge, and animals evolved the capability to exploit it. The chemical arms race between predators and prey, which in the modern world produces some of the most elaborate and toxic natural compounds known to chemistry, also has its roots in the Cambrian. While the chemical defenses of Cambrian organisms are not directly preserved in the fossil record, the evidence of intensive predatory pressure provides the evolutionary context in which the development of chemical defenses would have been strongly selected for. Modern marine invertebrates, many of which are direct descendants of Cambrian lineages, produce an extraordinary diversity of defensive chemical compounds, from the neurotoxins of cone snails to the skin toxins of nudibranchs to the ink of cephalopods. The selective pressure that drove the evolution of these chemical weapons was ultimately the same selective pressure that Anomalocaris and its relatives applied to Cambrian marine communities. The toxins and repellents of the modern marine world are the distant legacy of the first predator. What emerges from this detailed examination of the Cambrian arms race is a picture of evolution operating at extraordinary speed and with extraordinary creativity. generating a diversity of defensive and offensive innovations in just a few million years that had no precedent in the prior 4 billion years of life on Earth. The Cambrian arms race was not just a military competition between predators and prey. It was an evolutionary engine, a mechanism for generating biological complexity at a rate that passive, non-predatory ecosystems cannot match. Every spiny trilobite, every enrolled defensive ball, every burrowing trace, every mineralized shell, every sophisticated compound eye, every chemical defense is evidence of natural selection working at full intensity, driven by the most powerful selective force in the history of animal life: the daily, immediate, survival critical pressure of being hunted. The Burgess Shale itself deserves extended attention because it is not merely an important fossil locality. It is one of the most philosophically significant deposits of rock on Earth, and the debate that has surrounded its interpretation since the 1980s has raised questions about the nature of evolution and the role of contingency in the history of life that are still unresolved and still generating productive scientific disagreement. Charles Doolittle Walcott, who discovered and collected the Burgess Shale in the first decades of the 20th century, was a scientist of extraordinary energy and productivity. But he was also a man of his time, shaped by the prevailing evolutionary assumptions of the early 20th century. When he looked at the Burgess Shale organisms, he saw the ancestors of modern animal groups. He interpreted each strange Cambrian form as an early version of something he recognized: a primitive worm, a primitive crustacean, a primitive echinoderm. He fitted the Cambrian organisms into a framework of orderly evolutionary progress toward the modern world, a framework that was intellectually comfortable and that required no fundamental revision of his understanding of how evolution worked. Walcott's interpretations stood largely unchallenged for more than half a century. Then, in the 1970s and 80s, Harry Whittington at Cambridge University undertook a complete systematic reassessment of the Burgess Shale fauna, working with graduate students Simon Conway Morris and Derek Briggs. And what they found when they looked at the fossils with fresh eyes and without Walcott's preconceptions was something far stranger and more disturbing than Walcott had been willing to acknowledge. Many of the Burgess Shale organisms did not fit into any modern animal phylum. They were not primitive versions of modern groups. They were representatives of body plans that had no modern descendants, evolutionary experiments that had run their course in the Cambrian and left no living legacy. Opabania, with its 5 eyes and its flexible frontal nozzle ending in a claw. Hallucigenia, which was initially reconstructed upside down because it was so unlike anything modern that researchers could not determine which side was up. Wiwaxia, covered in scales and spines with a feeding apparatus, unlike anything else in the animal kingdom. These were not ancestors of modern animals. They were evolutionary dead ends, body plans that had diversified in the Cambrian and then gone extinct, leaving no descendants. The philosophical implications of this interpretation were developed most influentially by the evolutionary biologist and writer Stephen Jay Gould in his 1989 book, Wonderful Life, which brought the Burgess Shale to wide public attention and argued, with characteristic eloquence and force, that the history of life is fundamentally contingent. That if the tape of life were rewound to the Cambrian and replayed, the result would be entirely different from the world we have today. That the animals that survived the Cambrian filter, the lineages that made it through to become the ancestors of modern life, did so not because they were superior or more advanced or more fit in any general sense, but because of luck. Because of the random accidents of survival and extinction that could easily have gone differently, producing a world of radically different organisms in which nothing remotely resembling a human being would ever have evolved. The Burgess Shale, in Gould's reading, was a monument to contingency, to the role of chance in shaping the history of life. Gould's argument was enormously influential and remains widely read, but it has been substantially challenged and qualified in the decades since Wonderful Life was published. Simon Conway Morris, one of the researchers whose work formed the basis of Gould's analysis, reached almost the opposite philosophical conclusion from the same fossil evidence. Conway Morris argued, and has continued to argue in a series of books and papers, that the history of life shows strong patterns of evolutionary convergence, that similar environmental challenges tend to produce similar evolutionary solutions regardless of starting point, and that the evolution of something like intelligence and complex cognition was not a lucky accident, but a near inevitable outcome of the processes of natural selection operating in a world of physical and chemical constraints. The compound eye, which evolved independently in multiple Cambrian lineages and has been independently reinvented dozens of times in the history of animal life, is Conway Morris's favorite example. If eyes are that easy to evolve, perhaps the trajectory of complex animal life is more constrained and more predictable than Gould's radical contingency model implies. This debate between contingency and convergence, between Gould's vision of life as a wildly unpredictable lottery and Conway Morris's vision of life as a constrained exploration of a finite set of viable body plans, is one of the deepest and most interesting debates in all of evolutionary biology, and Anomalocaris sits at its center. Because Anomalocaris is, simultaneously, an example of an apparently unique Cambrian body plan with no modern equivalent, supporting Gould, and an example of the independent evolution of sophisticated predatory adaptations including compound eyes and grasping appendages that appeared convergently in multiple lineages, supporting Conway Morris. The first predator is both a monument to evolutionary contingency and an argument for evolutionary determinism, depending on which features you choose to emphasize. That ambiguity is not a weakness in the science. It is an accurate reflection of the genuine complexity of evolutionary history. The radiodont group to which Anomalocaris belonged was considerably more diverse than the single famous species name suggests. And the exploration of that diversity over the last 3 decades has added important nuance to our understanding of what the first predators were actually doing in the Cambrian Ocean. Anomalocaris canadensis is the type species, the one named first and the one most people mean when they use the name Anomalocaris. But it was far from the only radiodont in the Cambrian seas. The group includes dozens of described species distributed across multiple genera, from the comparatively gracile Amplectobelua of the Chengjiang fauna, with its distinctive frontal appendages bearing large raptorial spines, to the massive Herdia victoria of the Burgess Shale, with its strange 3-part anterior carapace that gave its head a bulldozer-like profile, to the extraordinary Lirarapax from China, to the recently described Titanosaurus guinea from the Marble Canyon locality in Canada. A radiodont with an enormous broad head shield that may have been adapted for scooping prey from the seafloor rather than pursuing it through open water. Among the most surprising discoveries in radiodont paleontology is the recognition that not all radiodonts were apex predators in the conventional sense. Several radiodont species from the Ordovician period, including Ediacaran ramskeldi from Morocco, preserved frontal appendages that were not the grasping, spine-bearing claws of an active predator, but instead a fan-like array of long, comb-like spines, clearly adapted for filter feeding rather than prey capture. Ediacaran was enormous, potentially reaching 2 meters in length, making it one of the largest animals of its time. And it was apparently feeding by swimming slowly through the water column, with its frontal appendage fans spread wide, straining zooplankton and small organisms from the water, in the same way that modern basking sharks, whale sharks, and manta rays filter feed today. The evolution of gigantic filter feeding from a predatory ancestor, producing an animal that looks anatomically like a predator, but feeds like a whale, is one of the most striking examples of evolutionary convergence in the fossil record. Modern baleen whales, basking sharks, and whale sharks all evolved their filter-feeding strategy from active predatory ancestors over the last few tens of millions of years. Ediacaran did the same thing from a radiodont predatory ancestor more than 460 million years ago. This convergence is remarkable, not just as a curiosity, but as evidence for the Conway-Morris view of evolutionary constraint. The ecological role of large, slow-moving filter feeder in a productive marine ecosystem is apparently a stable and attractive niche that natural selection finds repeatedly in lineage after lineage, separated by hundreds of millions of years of independent evolution. The physics of filtering large volumes of water for small prey imposes specific constraints on body shape and appendage morphology, and those constraints produce similar solutions regardless of starting point. Ediacaran solved them in the Ordovician. The whale shark lineage solved them in the Cenozoic. The solutions are not identical, but they are recognizably convergent. And the convergence tells us something important about the repeatability of evolution when the ecological opportunity is clear and the physical constraints are well defined. The Chengjiang fauna of China deserves particular mention as a complement and counterpart to the Burgess Shale, because it preserves a slightly earlier snapshot of Cambrian life and has yielded some of the most important radiodont specimens ever found. Chengjiang, in Yunnan province, was deposited approximately 520 million years ago, roughly 10 million years before the Burgess Shale, and it contains a fauna of similar diversity and exceptional preservation. The Chengjiang radiodonts include some of the most complete and detailed Anomalocaris specimens, known, with preservation of the eyes, the frontal appendages, the mouth apparatus, the lateral fin lobes, and, in some cases, internal structures that are almost never preserved. The Chengjiang fauna has also yielded evidence of the earliest chordates, the animal group to which we belong, including the famous Yunnanozoon and the slightly younger Hyquithis, tiny fish-like animals that were apparently prey items in the Cambrian ecosystem, and that represent the earliest members of our own evolutionary lineage. This is a fact worth sitting with. In the Cambrian Ocean where Anomalocaris hunted, there were also tiny, soft-bodied fish-like animals that were almost certainly prey. The earliest members of the vertebrate lineage, the ancestors of every fish, every amphibian, every reptile, every bird, every mammal that has ever lived, were small, vulnerable, probably not particularly fast, and living in an ocean dominated by the most effective predator of their time. The vertebrate lineage survived the Cambrian, clearly because we are here, but it survived as prey. The selective pressure of being hunted by Anomalocaris and its relatives was part of the evolutionary environment that shaped the early vertebrate body plan, that drove the development of a muscular, streamlined body for fast swimming, that selected for sensory systems capable of detecting approaching predators, that may ultimately have contributed to the evolution of the vertebrate brain as a sophisticated threat detection and evasion planning organ. We are, in a very real sense, the descendants of Anomalocaris' prey. Our brains, our senses, our capacity for fear and vigilance and rapid threat assessment, these are, at least partly, the evolutionary legacy of having been hunted by the first predator for millions of years in the Cambrian Ocean. The connection between Anomalocaris and the broader arthropod evolutionary tree is a subject of active research and some controversy. But the current consensus places radioodonts as a stem group relative to the true arthropods, meaning they are more closely related to arthropods than to any other animal group, but branched off before the evolutionary innovations that define the arthropod crown group, including the fully fused and articulated exoskeleton and the biramous limb structure that characterize true arthropods. This placement makes radioodonts enormously important for understanding arthropod evolution, because they preserve in visible anatomical form the intermediate stages between the segmented appendage-bearing body plan that radioodonts share with arthropods and the specific features of the arthropod exoskeleton that would eventually make that group the most species-rich animal phylum on Earth. The arthropods, the group that includes all insects, spiders, crustaceans, millipedes, centipedes, and their relatives, contain more described species than all other animal groups combined. There are more species of beetle alone than there are species of plant. The arthropod body plan, with its segmented body, its jointed appendages capable of being modified for a limitless variety of functions, from swimming to flying to silk spinning to venom injection, and its exoskeleton that can be hardened to almost any degree of rigidity, has proven to be one of the most versatile and successful blueprints in the history of life. And the origin of that blueprint, the evolutionary lineage from which the arthropod body plan emerged, runs through or very close to the radiodonts. Anomalocaris is not the ancestor of all arthropods, but it is a close relative of that ancestor. It is, in phylogenetic terms, something like an uncle, a collateral relative that illuminates the early evolution of a body plan that would eventually dominate terrestrial and marine ecosystems for the next 500 million years. The eyes of Anomalocaris, those remarkable compound eyes with their thousands of individual optical units, are particularly interesting in this context, because the compound eye is one of the defining innovations of the arthropod lineage, and its evolution in the Cambrian is one of the most significant sensory innovations in the history of animal life. Andrew Parker, a biologist at Oxford University, has proposed what he calls the light switch theory of the Cambrian explosion, arguing that the evolution of the image-forming eye was the trigger for the rapid diversification of animal body plans in the Cambrian. In Parker's model, Once one lineage evolved the ability to see other animals as spatially resolved objects, rather than merely as shadows or chemical gradients, the entire ecology of the ocean was transformed. Predators could see prey. Prey could see predators. The evolutionary arms race of predator and prey detection was initiated, and the explosive diversification of body plans that we see in the Cambrian fossil record was the result of that arms race playing out in evolutionary time. Parker's hypothesis is contested, as most strong evolutionary hypotheses are, and alternative explanations for the timing and mechanism of the Cambrian explosion include changes in ocean chemistry, increases in atmospheric oxygen, the breakup of the late Proterozoic supercontinent Rodinia, and the associated creation of new shallow marine habitats, and various developmental biology arguments about the evolution of the genetic toolkit that controls body plan development. The truth is probably a combination of multiple factors, as is usually the case with complex historical events. But whatever the full explanation for the Cambrian explosion, the evolution of the image-forming eye, exemplified most spectacularly in the Cambrian by the extraordinary eyes of Anomalocaris, was both a product of and a driver of the rapid ecological transformation of the Cambrian ocean. Fear, as a neurological and behavioral phenomenon, is among the most ancient and most conserved features of animal psychology. The neural circuits that generate fear responses in a modern mammal, including the amygdala and associated limbic structures, have deep evolutionary roots that trace back to the earliest vertebrates. And the earliest vertebrates were Cambrian animals living in an ocean with Anomalocaris. The connection is not merely poetic. Natural selection acts on survival, and survival in a predator-prey structured ecosystem requires the capacity to detect, assess, and respond to predatory threat. The organisms that survived the Cambrian predatory environment were those with the best threat detection capabilities, the fastest escape responses, the most effectively calibrated fear responses. We are the descendants of those survivors. And the predator that generated the selective pressure for those fear responses, that essentially invented the ecological context in which fear became a survival critical capacity, was Anomalocaris. This is perhaps the most profound and least comfortable insight that the study of Anomalocaris offers. When you feel afraid, when the ancient, wordless bodily sensation of threat activates the deep structures of your nervous system and floods your body with the physiological cascade of the fear response, you are experiencing something whose evolutionary roots stretch back 520 million years to an ocean in the Canadian Rockies and the hills of Yunnan province, where a large, compound-eyed, fin-swimming predator was pursuing soft-bodied animals that had never encountered anything like it before. The fear response is not a modern development. It is not a mammalian innovation. It is not even a vertebrate specialty. It is one of the oldest features of complex animal neurology, because the selective pressure that made it adaptive, the pressure of being hunted, has been present since the Cambrian. Anomalocaris did not last forever. The radiodont group appears in the fossil record from the early Cambrian, reaches its peak diversity in the middle Cambrian, and then declines through the Ordovician and into the Silurian, with the last known radiodont fossils dating to the early Devonian, around 400 million years ago. The reasons for the decline of the radiodonts are not entirely clear, and the pattern is complicated by the incompleteness of the fossil record and by the genuine difficulty of determining whether absence of fossils reflects true extinction or simply failure of preservation. What is clear is that by the time the great Paleozoic marine ecosystems of the Devonian and Carboniferous had fully developed, with their diverse fish fauna and their complex invertebrate communities, the radiodonts had been replaced as top predators by other groups, particularly the increasingly sophisticated and heavily armored fishes of the Devonian, culminating in the extraordinary placoderms, armored jawed fishes that reached lengths of several meters and were the dominant marine predators for much of the Devonian. The transition from Anomalocaris to the placoderms to the sharks to the modern marine predator community is not a simple story of one predator replacing another in a fixed ecological role. It is a story of escalating sophistication, with each successive dominant predator group bringing new anatomical and behavioral innovations that made it more effective than its predecessors, and each escalation of predatory capability driving corresponding escalations in the defensive and evasive capabilities of prey. The shark, which appeared in the early Devonian and has persisted essentially unchanged in its basic body plan for 400 million years, is in some sense the ultimate expression of a predatory design philosophy that Anomalocaris introduced in the Cambrian. A streamlined, fast-swimming, visually guided predator with effective prey capture structures and the sensory sophistication to find and track prey in three-dimensional water. The implementation is entirely different. The design philosophy is continuous. Every time you look at a hawk spiraling above a field and feel the aesthetic recognition of a perfect predatory machine, supremely adapted for finding and catching prey, you are looking at an evolutionary solution that began its development 520 million years ago. Every time a fish in a reef freezes at the shadow of a larger fish passing overhead, that freezing behavior, that ancient reflexive response to predatory threat, is the product of an evolutionary history that began when the first predator appeared in the Cambrian Ocean. Every time a rabbit bolts from a hawk, every time a deer freezes in alarm, every time a small invertebrate contracts into its shell at the approach of a threat, the behavioral script being executed was written in the Cambrian by the evolutionary pressure of being hunted by something with compound eyes and grasping claws, and the first mouth in the history of life designed specifically to crush living prey. There is a quarry in the Canadian Rockies, high in the mountains of British Columbia, where the rock is three-dimensional time, where the ocean of 520 million years ago is preserved in a layer of fine-grained shale no thicker than a good novel. In that rock, Anomalocaris canadensis is preserved. Its frontal appendages, those structures that White Eaves named as a separate shrimp species in 1886, are there. Its circular mouth, which Walcott thought was a jellyfish, is there. Its compound eyes, with their thousands of individual optical units, are there, staring out of the rock at anyone willing to look carefully enough. And in the same rock, trilobites with their own compound eyes and their calcified protective exoskeletons, and soft-bodied worms and early chordates, and a dozen other forms that represent the first complex marine ecosystem in the history of the planet. When you look at the fossil of Anomalocaris, what you are looking at is not a curiosity from an alien past. You are looking at the beginning of a story that has never ended, the story of predator and prey, of hunter and hunted, of the evolutionary arms race that has been running without pause for 500 million years, and that has produced, among its many consequences, the nervous system you are using right now to process these words. The fear response encoded in your amygdala, the eyes with which you are reading, the intelligence with which you are understanding. Anomalocaris did not know it was starting anything. It was simply hunting, with the best eyes and the best claws, and the best mouth that 500 million years of prior evolution had produced, in an ocean that had never seen anything like it. It could not have known that in doing so, it was setting in motion a chain of evolutionary consequences that would eventually produce, among countless other things, the species capable of finding its fossil, assembling its scattered fragments into a coherent whole, and recognizing in its alien face the distant origin of something deeply familiar. The three fossils that confused scientists for nearly a century. The shrimp that was not a shrimp, the jellyfish that was not a jellyfish, the mystery organism that contained both of them and everything else besides. When they were finally assembled into a single complete animal, what scientists saw was the first predator, the first hunter, the first organism on Earth that had been built, from compound eye to grasping claw to crushing mouth, for the sole purpose of finding, catching, and consuming another living animal. And in that recognition, if you are willing to follow the chain of consequence to its end, you see something that is not just paleontology. You see the origin of fear. You see the origin of the arms race that produced intelligence. You see the beginning of the long and violent and creative process that eventually, after 500 million years of escalation, produced a mind capable of looking back at its own origins and asking, for the first time in the history of life, where did I come from? And finding in the black shale of the Canadian Rockies something that looks back,